A new, error-corrected method for detecting cancer from blood samples is much more sensitive and accurate than prior methods and may be useful for monitoring disease status in patients following treatment, according to a study by Weill Cornell Medicine and New York Genome Center investigators. The method, based on whole genome sequencing of DNA, also represents an important step toward the goal of routine blood test-based screening for early cancer detection. In the study, published April 11 in Nature Methods, the researchers benchmarked the cancer detection performance of a new commercial sequencing platform from Ultima Genomics. They demonstrated that a low-cost platform such as this one enables a very high depth of coverage, a measure of the sequencing data quality, allowing investigators to detect extremely low concentrations of circulating tumor DNA. Adding an error-correcting method greatly improved the accuracy of the technique. We're now entering an era of low-cost DNA sequencing, and in this study, we took advantage of that to apply whole genome sequencing techniques that in the past would have been considered wildly impractical, said senior author Dr. Dan Landau, the Bibliovich Family Professor of Medicine, and a member of the Englander Institute for Precision Medicine and the Sandra and Edward Meyer Cancer Center at Weill Cornell Medicine, and a core faculty member of the New York Genome Center. Blood test-based, liquid biopsy, technology for early cancer detection and monitoring of cancer burden in patients could revolutionize cancer care. However, sensitively and accurately identifying the mutational signatures of cancer, just from tiny concentrations of tumor DNA in blood samples, has involved major challenges. The Landau Laboratory for most of the past decade has been working to overcome these challenges using methods based on whole genome sequencing, not just targeted sequencing of stretches of DNA where mutations are. In a study published last year, they showed that they could reliably detect advanced melanoma and lung cancer from patient blood samples, even without access to sequence data from tumor samples. In the new study, they took their approach a step further. First, they showed that the low cost of a new sequencing platform enables a depth of whole genome sequencing that would have been prohibitively expensive with older technology. Using that platform alone, and having the known mutational patterns in patient tumors as a guide, they were able to detect tumor DNA in patient blood samples at concentrations in the part per million range. All samples in the study were collected after obtaining informed consent from the patients. Next, the team enhanced the accuracy of this approach with an error correction method that makes use of the redundant information in natural two-stranded DNA. They showed that the combined technique has extremely low error rates, making it feasible in principle to use on blood samples without also needing access to patient tumors. Collaborating with other research teams, the researchers demonstrated the potential of this high-sensitivity, low-error approach by using it to detect and assess very low cancer levels in patients with bladder cancer and melanoma from blood samples alone. This collaboration allowed us to analyze circulating tumor DNA from patients with bladder cancer and identify the distinct mutational signatures that my lab has extensively studied, said Dr. Bishoy M. Faltas, Chief Research Officer of the Englander Institute for Precision Medicine and Associate Professor of Medicine and of Cell and Developmental Biology at Weill Cornell Medicine. Faltas is also a urologic oncologist at New York Presbyterian/Weill Cornell Medical Center. Incorporating these signatures into the analysis significantly increased the sensitivity of circulating tumor DNA. We were able, for example, to see increases in circulating tumor DNA levels after treatment in patients with cancers that progressed or recurred, and declines in those levels in patients whose cancers had full or partial responses, said first author Dr. Alexander Cheng, a postdoctoral researcher in the Landau Laboratory during the study. These results allow us to think about a future in which we can detect and track cancer from blood tests alone, said Dr. Landau, who is also an oncologist at New York Presbyterian/Weill Cornell Medical Center. Investigation of the paper, error-corrected flow-based sequencing at whole genome scale and its application to circulating cell-free DNA profiling, differentiating sequencing errors from true variants is a central genomics challenge, calling for error suppression strategies that balance costs and sensitivity. For example, circulating cell-free DNA, cfDNA, sequencing for cancer monitoring is limited by sparsity of circulating tumor DNA, abundance of genomic material in samples and pre-analytical error rates. Whole genome sequencing, WGS, can overcome the low abundance of cfDNA by integrating signals across the mutation landscape, but higher costs limit its wide adoption. Here, they applied deep, 120 times, lower cost WGS, Ultima Genomics, for tumor-informed circulating tumor DNA detection within the part per million range. They further leveraged lower-cost sequencing by developing duplex error-corrected WGS of cccFDNA, achieving 7 times 10⁻⁷ error rates, allowing us to assess disease burden in individuals with melanoma and urothelial cancer without matched tumor sequencing. This error-corrected WGS approach will have broad applicability across genomics, allowing for accurate calling of low-abundance variants at efficient cost and enabling deeper mapping of somatic mosaicism as an emerging central aspect of aging and disease. Ultima and Illumina sequencing datasets of human-mapped reads in mouse PDX datasets, N equals 3. A homopolymer size estimation of bases between two PCR duplicates, all samples combined, in Ultima datasets. B. Homopolymer size estimation of bases between a read and the aligned reference, all samples combined, in Ultima datasets. C. Homopolymer size estimation of bases between two PCR duplicates, all samples combined, in Illumina datasets. D. Homopolymer size estimation of bases between a read and the aligned reference, all samples combined, in Illumina datasets. E. Indel calling accuracy by PCR duplicate family sizes in Ultima datasets, N equals 3 in each box plot. F. Indel calling accuracy of Illumina sequencing reads, for single family reads, N equals 3 in each box plot. G. Frequency of homopolymer sizes across the human genome. For box plots in E and F, the lower and upper ends of boxes represent the 25th and 75th percentiles of the data, respectively, and the horizontal lines represent the median. The whiskers represent at most 1.5 times the IQR. Accuracy in E and F is defined as the number of correct homopolymer assignments in individual sequencing reads divided by the occurrences of that homopolymer size in the human genome in all sequenced. Flow-based sequencing provides predictable error-robust motifs. A single nucleotide variant analysis of matched Ultima and Illumina sequencing datasets across 96 trinucleotide contexts. Cycle shift motifs, described in B, are indicated by plus signs. B left, example sequencing of a TGC trinucleotide in flow space. Given a flow order of T greater than G greater than C greater than A, one full flow cycle of each nucleotide should provide a 1 greater than 1 a 1 greater than 0 signal. Top right, example of how a T, G greater than A, C alt disrupts the cycles in flow space base calling. 2 sequencing cycles are required to fully resolve a TAC sequencing motif. We refer to these types of motifs as cycle shift motifs. Bottom right, example of how a T, G greater than C, C variant does not affect the cycles of flow space base calling. See error rates in Ultima and Illumina sequencing datasets for trinucleotide variants that alter the flow space sequencing cycle, N equals 120 in the cycle shift motif box plots, blue, corresponding to the 40 trinucleotide variants that are classified as cycle shift motifs across 3 mouse PDX plasma samples. N equals 168 in the non-cycle shift motif box plots, red, corresponding to the 52 trinucleotide variants that are not classified as cycle shift motifs across 3 mouse PDX plasma samples. P-values were measured using a two-sided Wilcoxon test. Error bars in A represent the standard error of the mean. For box plots in C, the lower and upper ends of boxes represent the 25th and 75th percentiles of the data, respectively, and the horizontal lines represent the median. The whiskers represent at most 1.5 times the trade-offs between deep targeted sequencing and modest whole genome sequencing for ctDNA detection. A mutational burden, number of SNVs, of 22 cancer types retrieved from the pan-cancer analysis of whole genomes consortium. The numbers along the x-axis represent the number of tumors analyzed per cancer type. B. Median ctDNA detection opportunities using a whole genome approach with 10x sequencing coverage, a 10-target panel at 10,000x coverage and a 1-target panel at 10,000x coverage. The pink shaded area represents tumor types for which targeting only a few sites may offer benefit over whole genome sequencing. The blue shaded area represents tumor types for which a whole genome approach will offer more opportunities to detect ctDNA over targeted panels. The lower and upper ends of the box plots in A represent the 25th and 75th percentiles of the data, respectively, and the horizontal lines represent the median. The whiskers represent at most 1.5 times the IQ. Circulating tumor DNA cost and coverage analysis between Illumina and Ultima sequencing in a matched sample. Areas under the curve, AUCs, are measured by calculating the area under a receiver operating characteristic curve comparing a given group, for example, Illumina 20X at 10⁻⁶ expected tumor fraction, to its platform and coverage-matched cancer-free control, for example, Illumina 20X, expected tumor fraction of 0. All AUCs at expected tumor fractions of 10⁻⁴ and greater were 1. Z-scores of a given sample are calculated against their coverage and platform-matched cancer-free control, expected tumor fraction of 0. Variant allele frequencies for variants across denoising approaches. Variant allele frequencies, calculated using unfiltered sequencing reads, in positions where a variant was found using UMI-agnostic denoised reads, single-strand corrected reads and in duplex corrected reads. Allele frequencies of 0, 2 and below are colored in red. Comparison of detected UV-derived mutations using duplex, single-strand and UMI-agnostic denoising methods. A cosine similarities by cancer stage at baseline time points, pre-treatment or pre-surgery, for UV and CH-associated signatures. B. Comparison of duplex, single-strand and UMI-agnostic denoising methods to detect melanoma-associated variants using a single-read variant calling pipeline for pre-treatment plasma samples from melanoma patients, top, and cancer-free controls, bottom. P-values were measured using a two-sided Wilcoxon test. For all box plots, the lower and upper ends of boxes represent the 25th and 75th percentiles of the data, respectively, and the horizontal lines represent the median. The whiskers represent at most 1.5 times the IQR. Tumor-agnostic copy number-based tumor fraction estimation in stage 3 and 4 melanoma and cancer-free control samples. Samples include cancer-free controls, N equals 10, stage 3 melanoma, pre-surgery, N equals 10, and stage 4 melanoma, pre-treatment, N equals 4. Dotted line at 0.03 represents the limit of detection of iCR-CNA. For box plots, the lower and upper ends of boxes represent the 25th and 75th percentiles of the data, respectively, and the horizontal lines represent the median. ctDNA dynamics throughout treatment in melanoma patients. A changes in circulating tumor DNA, increase or decrease, relative to the earliest sample time point. Solid lines represent patients with recurrence or progressive disease, and dashed lines represent patients with either partial response or who are recurrence-free following treatment. Closed and open circles represent samples with and without detected ctDNA, respectively. B. Difference in ctDNA relative to the pre-treatment time point stratified by clinical outcome. One sample did not have a pre-treatment time point available, MEL 15, progressive disease, and so a day 9 post-treatment time point was used as baseline. For box plots in B, the lower and upper ends of boxes represent the 25th and 75th percentiles of the data, respectively, and the horizontal lines represent the median. The whiskers represent at most 1.5 times the IQR. P-values were calculated using a two-sided Wilcoxon test. Major signature contributions from urothelial cancer patients' tumors measured through whole genome sequencing. Top, total mutation counts per sequenced tumor. Bottom, signature contributions. Trinucleotide frequencies were fit to the entire COSMIC database, version V. When a patient had 2 or more tumors, B01, B04, B15, B16, B17, B18, B19, we measured signature contributions of mutations that were present in 2 or more tumors and thereby likely reflect mutations that arise earlier in tumor evolution.